This is our presentation on emotional arousal and its effect on cortex activation by Brooke Shantz, Lynn Pham, and Jonathan Baker. The study we're going to present is called Emotional Arousal and Activation of the Visual Cortex, an fMRI analysis. It was researched by Peter J. Lang, Margaret M. Bradley, Jeffrey R. Fitzsimmons, Bruce N. Cuthbert, James D. Scott, Bradley Mulder, and Vijay Nangia. The purpose of this study was to examine the emotional response in the brain through looking at images intended to cause emotion in people. The researchers wanted to see where functional activity specifically occurs when people view emotional images. What do you need to know to understand this study? There are some key elements important to understanding it. The first is a previous study done in 1997 by Richard Lane and his colleagues at the University of Arizona. I'll speak more about this study in just a moment. To understand the study, it was also helpful to understand how to read an fMRI scan, as well as the purpose of fMRI studies. As we know, they're used to measure blood flow in the brain, and it's also extremely relevant to understand what it means when there is more blood flow or more activation in certain areas of the brain than others. In Richard Lane's study, he aimed to find the neural substrates, or parts of the nervous system underlying pleasant and unpleasant emotion. They predicted that pleasant and unpleasant activation would result in increased activation in the thalamus and medial prefrontal cortex. They also predicted greater cortex activation in both emotion conditions relative to the neutral conditions. They also believed the amygdala would show more activation in response to the unpleasant compared to pleasant emotion conditions. They showed participants photos from the IAPS, or the International Affective Picture System. They used positron emission tomography to produce 3D images of the function occurring in the subjects' brains. Subjects were 12 women. The most relevant finding of their study was that emotional images in general cause greater brain activation than neutral ones. Their results also showed that significantly greater blood flow was observed in the visual cortex of subjects for unpleasant pictures than for neutral images. There were no observable differences in the activation between neutral and pleasant stimuli. Another study which provided good background information for looking at this study was done by Jean Royet and colleagues who tested regional cerebral blood flow based on how pleasant or unpleasant a stimulus was. They presented a stimuli to participants using all sensory modalities. Emotionally valenced or heavily emotional stimuli led to the increase of regional cerebral blood flow. It was also shown that pleasant and unpleasant emotional judgment recruit the same network in the left hemisphere. Only emotionally valenced olfactory stimuli, or stimuli using your nose, induced bilateral regional cerebral blood flow increases in the amygdala. The third and last study we used to gain some background knowledge on the subject was done by Vishnu Murthy and his colleagues at Duke University. Similarly, he studied emotion and localization. However, his interest lied more in the area of the localization of emotional memory. He wanted to characterize the neural systems that have been consistently associated with emotional memory formation. They looked at memory encoding and the effect of emotional versus neutral stimuli and what effect they had on encoding and the function of the amygdala in encoding. The material in our main study of focus has relevance to what we have learned in class. The study we focused on showed participants images that researchers categorized as pleasant or unpleasant. The reason participants were intended to have these reactions is due to functioning of their cognitive processing systems. The study can actually be representative of top-down or bottom-up processing. It could be top-down because we have previous ideas and experiences related to the emotions that each picture is intended to evoke. It could be bottom-up processing because participants aren't told they're going to be presented with these photos, and when presented to them, they begin processing them. How did the researchers obtain their results? Well, finally we'll get into how this particular study was done. It was performed in 2 phases. The first phase had a checkerboard flash in front of participants in the right and left and center visual fields while monitoring their brain activity. This was essentially to check that the correct hemispheres were being activated in participants. In phase 2, participants were shown 3 sequences of 8 images, each for 12-second intervals, with 12-second intervals also in between as just blank screens. The images presented to participants were either pleasant, unpleasant, or neutral. Pleasant images were pictures of things like laughing babies or intimate couples. Negative images were things like violence or mutilated bodies. Neutral images were things like cooking utensils or people with neutral expressions on their face. Within this phase, 2 experiments were conducted. One which tracked blood flow in the brain, which was the fMRI study, and one which tracked eye movements and skin conductance. Here you can see the fMRI results. You can see the areas of functional activity lit up in the brain. To help display the information best, functional maps were averaged over all participants and then overlaid on a representative anatomical image. The results. Researchers found that emotional and neutral images differ in the extent of functional activity in the visual system, as shown in the fMRI scans. The right occipital gyrus activated only when emotional pictures were shown. Emotional stimuli showed greater activation in visual cortical areas than the neutral images did. Another interesting finding of this study was related to a difference among genders. Researchers found that in the right hemisphere, women showed significantly more brain activity for unpleasant than for pleasant stimuli. Men, however, showed significantly more activation for pleasant than for unpleasant stimuli in the right hemisphere. However, in terms of increased functional activation from emotion, it's generally the same for men and women. The following graph helps to visualize this gender difference from their findings. The authors found that their research was consistent with what they had previously found in other studies of emotion and anatomical localization. Their findings of gender differences were also consistent with previous findings. They felt that their findings related to the importance of psychophysiological measures in mapping the brain. What did the authors do well? Prior to the main portion of the study, the researchers devoted a part of their experiment to checking that participants' brain regions activated correctly upon flashing a neutral stimuli in each visual field. This helped to provide a base estimate of how reactive they were to visual content without any emotional meaning attached to it. This seemed like a really good way to assure accuracy later in the experiment.